All right, so we're off. Hey, everybody. Super excited to be diving into our first Pearl related fireside chat, where we'll be focusing in on your questions, questions from our recent Pearl webinar, questions following a recent Optispan podcast that covered our Pearl trial results. And there was a lot of different questions, a lot of them revolving around the bioavailability of rapamycin, dosing, and generally a lot of people interested in their own rapamycin journey and the clinical effectiveness of rapamycin, what they should be paying attention to, and how they should be engaging with rapamycin in order to, sort of optimize their experience and the results they may be getting down the line. People really interested in learning about, the learnings that we got from the Pearl trial and how we plan to carry that forward. So excited to jump into a bunch of your questions. So I think I'll just kick it off with the first question. I just want to actually first off introduce, again, no introductions needed. Dr. Z, if you want to give your short introduction leading into this, and then we'll go to Stefanie and dive into the questions from there.
Sure. Well, thanks for being on the call here. My name is Sajad Zalzala, Dr. Z for short. I am the co-founder and chief medical officer of AgelessRx. I have a strong interest in longevity medicine, functional medicine, which kind of was what drove me to start AgelessRx. It'll be 5 years ago now. I have extensive telemedicine experience, and I'm also the clinical researcher on the Pearl trial and the PI and a bunch of other trials that we have upcoming, we've done and we have upcoming. So I'm excited to be able to answer some of these questions for the community and provide more education and clarification on some of these questions.
Fantastic. And on to Stefanie.
Yeah, I joined Ageless just a few months ago, but it's been a really exciting chapter to join in. I'm the VP of the Research and Applied Science, so overseeing a lot of the efforts on the ground that the team is working on.
Yeah, and I'm Girish Harinath. I started working at Ageless back in 2023, and super excited about the directionality that we're going. Really diving into the clinical effectiveness and really personalized applications even of these different gerotherapeutic candidates. So excited to really get into our question and answers from the Pearl trial. So let me kick this off with question number one. And I think this is very relevant. What would you say are the most noteworthy results from the Pearl trial? We looked at a lot of different things. We looked at safety and different parameters of clinical effectiveness, different what we can call health span metrics. So what were the most noteworthy results, and what are the implications in terms of how you're planning on following up on the data that you got from this trial?
Yeah, great question. So, as we mentioned before on the previous Pro webinar, and by the way, if you haven't seen that, you should probably stop, go watch that, and then come back, because we're going to be referencing that webinar quite a bit, and that was like an hour and a half, including the Q&A. As was mentioned in the webinar, the results webinar, we were looking mostly for kind of safety. Is rapamycin at these intermittent doses safe enough for healthy populations? There really wasn't very much data, and so that's why we kind of launched the trial. So I think that's kind of the most noteworthy of all is this appeared to be safe at the doses that we studied, in the time frame that we studied it, in the population that we studied. We did not see any major adverse reactions or any cause for concern when it comes to safety. So I think that's like the big takeaway from the Pearl trial was, this rapamycin appears to be safe enough to move forward on as to study further. So that, I think that's the biggest implication of the trial, and that was our primary goal, to be honest. I mean, if we got other data from the trial, that that's all icing on the cake, and in my opinion. The fact that we saw some measurements of muscle lean tissue increase on DEXA scan. I think that was unexpected, to be quite honest. But very noteworthy, I think, because there's a lot of people who think that manipulating mTOR could affect muscle mass in a bad way, and I think this study, even though small and very modest, seemed to indicate that, hey, look, there's actually positive benefits here. So I think those are kind of the biggest noteworthy in my mind. And then we saw hints of quality of life improvement. Again, I mean, we ended up underdosing, I think, for a majority of our participants, especially our male participants. So the question the question that needs to be asked is like, would higher doses help male participants, male patients? I think the answer is yes. But like I said, I think the most noteworthy is we can move on to study some of these questions.
Yeah, fantastic. And I don't know if there's any, I'll add a quick addition in terms of what really excited me about some of these trial results is number one, what you mentioned, Dr. Z, the unexpected results, because we weren't really powered for this, was seeing improvements in lean muscle mass, specifically in women, even though it was a smaller cohort of women. And I think that is noteworthy, as you said, because there's been a lot of, I think, talk in the community surrounding a few papers that came out with like a single administration of rapamycin showing that uh, it adversely altered branch chain amino acid metabolism in muscle. And then the community kind of took that, or certain individuals did, suggesting that, based on how mTOR behaves in muscles, based on its role, based on the study, maybe it's, adversely affecting muscle mass in individuals and showing an improvement in muscle mass. And I think we saw somewhere around a 5.6% mean improvement within the individuals taking 10 milligrams compounded rapamycin, specifically women. That was really postmenopausal women, I should add. That was super interesting, and I don't want to forget also the improvements in pain that we saw, which seems to be, I don't know, correct me if I'm wrong. It seems to be one of the most common anecdotal experiences from individuals is self-reported improvements in pain.
Yeah. We definitely heard that, from many people, including Matt Kaeberlein himself. I think that was the reason that got him going on rapamycin. With the story of frozen shoulder, he talks about several times, on across many podcasts. I mean, I think one of the reasons why we saw that was because, pain is an issue as you get older, and I think it's very tangible, and I think it's what a lot of people worry about. So I wasn't surprised there. I was a little disappointed we didn't see a stronger effect. But I think we would if studied for either a longer period of time or at a higher dose.
Yeah. Welcome. I think Stefanie, you might have the next question or follow-up.
Yeah, fire away. Question 2. Let's go.
Yes. So for the for the bioavailability of compounded rapamycin, what are the implications of lower doses? And what are the advantages of taking the compounded or non-encapsulated rapamycin for the patient?
Sure. Like we mentioned in the Pearl webinar, we didn't know going into the Pearl trial. It didn't show up on anybody's radar that we consulted with or any of the trials that we looked at. We assumed that all forms of sirolimus or rapamycin were kind of equivalent. It wasn't until later that somebody pointed out that there was this encapsulation of the Pfizer had encapsulated their sirolimus rapamycin to enhance the absorption. And so we realized in another trial, and again we did a webinar dedicated to that, so I don't want to rehash that it turned out to have a lower absorption. We think somewhere in the neighborhood of about like 1/3. So you need to take basically 3 times as much. So what's the implication? So the implication was, even though we thought we were dosing at 5 milligrams and 10 milligrams, kind of the kind of the standard dose, 5-6 milligrams is what was thought to be standard. And then we thought we were doing a higher dose than that because some of our advisors recommended that we test a higher dose. In reality, it turned out we had like an almost almost non-existent, not non-existent, but like a very, probably too low. The 5 milligram compounded was probably too low to make any difference. and then the 10 milligram turned out to be about maybe 3.3, 3 milligrams, 3.3, which are starting to get closer to that 5 to 6 milligrams, which is, so that's the implication there, is that, I mean, drugs require certain levels and, area under the curve and, things like that in order to work. I use the example of Tylenol. You can't take 50 milligrams of Tylenol and expect it to work for your headache. You need to take 500 or 1,000, So same sort of thing. So that's kind of the implications of a lower dose, is just we probably just didn't see the responses we were hoping for because of lower dose. Now, in terms of the advantage, again, I talk about this on the other podcast, but just to really quickly kind of go over it. First of all, I mean, a placebo-controlled trial is almost impossible without using the compounded form. Other people are trying and finding a lot of difficulty just because of the way that the Rapamune and the Dr. Reddy's generic Reddy's generic. Just the way it's the shape, the size, the number of pills you have to take. It only comes maximum 2 milligrams. It would have been impossible for us to actually use the encapsulated form and do a placebo. So that's one thing from a clinical trial perspective. The other thing from a clinical perspective, I mean, compounding allows you to put whatever dose you want in there. You just ask the pharmacy. So it's easier to take, in my opinion, because it's only one capsule or one tablet, and you could adjust the dose whatever you want. As a clinician, we're not bound to the 0.5, 1, and 2 milligram tablets that are commercially available. We could do 10.25 milligrams, for example, or 18.2, whatever the dose turns out to be optimal. What is optimal is a different story altogether. But again, I'm just thinking about what the possibilities are. And then a couple other things. At the cost, it seems to be more cost competitive, the compounded version versus the commercial version. And also commercial drugs, especially generic commercial drugs, tend to be prone to supply disruption. So, there have been many times, even in the last like 5 years, that I've seen like generic drugs just the manufacturer just decides they don't want to make it, or they have a problem with their plant, because a lot of these medications are made in other countries, India, China, etc., and we import them. If there's an importation problem, or if there's a quality control issue with the plant, a contaminant, then the whole supply chain just shuts down. Whereas the compounded, they have different ways of getting the raw materials for the rapamycin to compound, so it's much less prone, in my opinion, in my experience, much less prone. Look at semaglutide, for example. I mean, they can't keep up with the demand, and so the compounding pharmacies are coming to the rescue because they can find other ways around the supply constraints. So I think ultimately that will probably be one of the biggest advantages of compounding. Is that and cost.
Yeah, fantastic. Thank you for that. And I think, one of the things that kind of like surprised me, as you mentioned, the lower dosages that we used. One thing that I think was truly novel that came from this trial is that most people aren't looking at that low doses of rapamycin within clinical trials. So we're seeing something equivalent to amino 3 milligrams of, encapsulated or generic commercial rapamycin. We can get into the lingo, but seeing effects, effectiveness even in lean muscle mass and pain with those low doses was interesting. And this probably also had something to do with the safety, relatively good safety profile that we saw within the trial as well. So all really interesting data to come out of this in my perspective. And just to like swiftly follow up on that, we're talking now, a lot of people are interested in the different formulations of rapamycin, different rapalogs. But like as we begin talking about this within the field more often, right? Compounded versus generic, what are the different formulations? How would you advise maybe talking about these different formulations? What's on your radar? And then, how can we confirm, some of our patients are wondering, how do I know that generic and compounded are equivalent, right? They're equally effective. Or some ways that you think about this strategically as a clinician?
Yeah. So, I mean, well, first of all, I'm going to kind of answer kind of the second part of the question here about the lingo around this. So, a lot of people, I mean, it's kind of all fault because we've used this lingo, compounded versus generic, or compounded versus commercially available, I think would be a little bit more accurate. The reason why we use generic version, and there's only 2 generic manufacturers as of as of September of 2024 that I know of that are on the market. The Dr. Reddy and a new one, and a relatively new one called Glenmark. I mean, I don't know how long it's been around, but I've just recently known about the Glenmark, and we'll talk about that a little bit later. But the reason why, we use the word generic because the Rapamune, the Pfizer branded Rapamune is prohibitively expensive. I think it'll be like thousands of dollars a month if somebody wanted to use that brand. So I mean that's kind of a non-starter for a lot of people. So the brand Rapamune is just not feasible for a lot of people. So that leaves us with generic, and then it leaves us with compounded version. But I think like more technically, in order to kind of like remove the whole, compounded versus manufactured, I think we can probably refer to it as non-encapsulated versus encapsulated. And I would say, non-encapsulated would be, compounded capsules, compounded tablets, or just, the raw powder form. Some people were talking on messaging board about getting raw powder form and measuring it out. That would be, I wouldn't recommend that, but that would be considered not all, kind of all non-encapsulated. There's no attempt by, to enhance the absorption of sirolimus or rapamycin. As opposed to encapsulated, then you start covering the commercially available stuff. The Rapamune for sure, the Dr. Reddy's we know for sure, maybe the Glenmark. I don't have much information about the Glenmark version at this point. And then also there's a liquid available. It's by Pfizer. There's a sirolimus liquid preparation that's supposed to be also kind of encapsulated as well. So I think all those would kind of fall under encapsulated. And then non-encapsulated would be like so encapsulated. They specifically put things. They manufacture the tablet, the liquid, the preparation to enhance absorption, whereas non-encapsulated means non-enhanced absorption. Now, what makes it a little bit messier is there are techniques where you can compound a capsule or a tablet that does enhance absorption, and we tested that out before, but I'm not going to go down that route because I don't want to add confusion to it. But I think it's best to use lingo encapsulated versus non-encapsulated. Anyway, in terms of like, I think the question was, what was the first part of that question again? I got really off track in terms of lingo.
Yeah, no, it's an important discussion, and
Yeah.
the lingo. I think the next part is individuals concerned or interested in, how do I decide which formulation is effective for me? How do I know my compounder rapamycin is, or on non-encapsulated rapamycin is as effective as the generic? Are there any ways that you're testing
Yeah,
that?
absolutely. Well, we talked about this during our webinar when we talked about the bioavailability paper, but just kind of give like a very quick overview of it. The best way seems to be checking a 24-hour level. So doing ingesting rapamycin, whatever form you're given or you have access to, and then checking your blood test drawing your blood 24 hours later, checking the sirolimus levels. And this can be done at LabCorp or Quest, probably like most standard labs could probably also do it, but LabCorp and Quest seem to be kind of the standard. And then in terms of the ranges, we talk a little bit about that in our bioavailability. It seems to be, we're not sure. I mean, again, I don't want to go into the details there, but if you target somewhere between maybe 2 and 5 nanograms, I think it's per deciliter or milliliter. Forget the units exactly, but 2 to 5 would be the number. Up to 10. Once you get beyond 10, I think you start getting a little too high on the dose. And then once you get below 2, I think that's too low on the dose. So between 2 and 5, maybe up to 10, we're not exactly sure yet, but it's something we're studying.
Stefanie, I think we might have the next question on you.
Yeah, I think we kind of covered question number 4 as part of that answer. So I'm going to go to the next question. So the person asked if you're going for a high Cmax with biweekly dosing, or should you do a lower dose weekly? So kind of just like what should your, and this question has come up a lot, what should your strategy be for taking rapamycin and why?
Right now, it's really anybody's guess. And well, the other thing is like when you say high Cmax, again, we talk about this in the bioavailability paper, but getting the, measuring the Cmax is very tricky. because of the pharmacokinetics of sirolimus, which is why we didn't bother, because it's a fool's errand unless you have somebody, you know, locked away in a lab and checking their blood every 10 minutes or something like that, which, you know, clinically impractical. So that's why we kind of settled on the 24-hour level, which seemed to provide a more stable. Again, more details in our bioavailability webinar. Honestly, we don't know. I mean, you know, it could be both. You know, you know, the unsatisfying answer, it could be both. You know, there are some disease conditions or some, you know, tissues that respond to a higher biweekly dosing, and there could be other mechanisms and tissues that, you know, that prefer a lower weekly dosing. You know, again, it's really early days with rapamycin and these sorts of questions. So maybe check back with me in about 5 years.
Yeah, great. I think that makes sense. And you know, it sort of is a call to action to the field to really start looking a little bit deeper at these different dosing regimens and associating them with clinical effectiveness markers so we can get more clarity. And I know that we're running some studies that are hoping to help get to the bottom of that as well. Another question that was you know, came up a few times during the webinar and from participants afterwards is, you know, where do you fall on the safety of higher doses of rapamycin, right? With the Pearl trial, we've said we're using, you know, relatively low doses compared to what the field is generally doing. People are really eager, like, you know, the safety results from, you know, what's equivalent to 3 milligrams of encapsulated rapamycin, we're pretty clean. It's encouraging. So, you know, 6 to 8 milligrams seems like something that people are very interested in and more comfortable doing after seeing these initial results. Like they want to take the dose a little bit higher. And, you know, is there a case for going up to 6 to 8 or even 10 milligrams in your opinion at this point in time? And how do you feel about, you know, we did see some early results with very low doses of rapamycin. Maybe if we extended the trial longer, there more things could have popped up. So yeah, how are you feeling on that right now based
Yeah,
on
I mean, so this kind of falls into speculation and kind of my personal slash professional opinion, you know, and some of it's based on available science, and some of it there's just not enough science to comment. But I'm kind of like, I'm going to pull out my crystal ball for a minute and give some speculation. This will be purely speculation, um, you know, and just kind of my knowledge of what I've learned so far in rapamycin. So there are some concerns about very high doses. I think if you look at the Joan Manick studies, I think they looked at the higher doses, and there was a higher incidence of side effects. So at some point, going too high is going to be detrimental. And my concern about pushing it too high for too long is, you know, you may not know the detrimental effects until like years down the line. So that would be my concern about people pushing the dosing too high, is that we currently don't have a good way to recognize when somebody's gone too far. So that's why for now I'm not recommending the high doses, because we don't have, you know, unlike, let's say, a statin medication, for example, where if you go too far, then you suppress your LDL too much, you know, and then you can back off on the dose. You know, if you go too far, then you start getting, you know, mitochondrial effects, myalgias, et cetera, and then you can back down on dose. Um, we don't currently have any reliable markers like that I'm aware of for rapamycin. So that would be my main concern. We don't have any like ceiling or some safety valve or anything like that for rapamycin right now. So for now, I'm not recommending a higher dose. You know, the highest I would currently safely recommend is 8 milligrams of the encapsulated form once a week, which is roughly equivalent about 25 milligrams of the non-encapsulated form. You know, could we see results? Could we see long-term benefits with a very, very low dose, like 1 milligram a week? I would say, I mean, if we didn't see it at 12 months, I would say there's a low chance that we would see it beyond that. You know, if we didn't see there any results at 12 months. So that's why I'm not optimistic about very, very low dose, like 1 or 2 milligrams per week. That's why I think the sweet spot is somewhere between 4 and 8 of the encapsulated, where maybe some patients do better with 4. Let's say female patients might do better with 4. Maybe male patients need 6 or 8. You know, again, that's just kind of speculation based on the data that are currently seen. But no, I would not push it too high or too low.
So just to keep going with the questions here, um, keeping with the dosing questions, a lot of people have asked us, there's a trend for intermittent dosing, like very intermittent dosing, where there's like 8 weeks on, 4 weeks off kind of thing. And Peter Attia is one of the people who advocates for this approach, which I think encourages a lot of the discussion. What are your thoughts about doing, is that necessary? Is it better? Is it worse? Do we know? Do you think, like, especially in light of some of the concerns around higher dosing, would it negate some of those concerns? This one says positive benefits along with recalibration of things like insulin for overactivation of mTOR2.
Yeah. Well, first of all, we didn't necessarily see any changes in insulin with the doses that we studied. And when I look at clinical data from patients who are prescribed this the 6 milligram a week dose through our telemedicine platform, I'm not seeing any insulin resistance. We do see some increases in A1C, but that's a whole different story altogether. It doesn't seem to be related to insulin resistance. It seems to be more about some kind of red blood cell or some kind of a phenomenon we don't yet understand. So I mean, if somebody is seeing insulin resistance with rapamycin, they're probably taking it a little bit too far. Is my guess. In terms of like cycling on and off, I mean, at this point, I think it's anybody's guess. There could be benefit to it. There couldn't be, there may not be benefit to it. I mean, one kind of, mark in favor of it is, when you look at the mice trials, for example, I think they took, I don't remember the exact details, but I think they put mice on it for like 12 weeks or so. And then they continued to have benefits throughout, for extended period of time. In humans, I don't know how long that extended period will be. So is there a case for maybe 12 weeks on, 12 weeks off? I think that's what some people like Matt Kaeberlein do, for example. But I think the reason why he starts it back up after 12 weeks is because that's when his shoulder starts aching again, and he knows hey, it's time to get back on the rapamycin. The problem with that is I mean, there could be you imagine, okay, if the inflammation in the shoulder, I'm sorry, I'm going to pick on you, Matt K, here for a minute, because you publicly, you made yourself a public target here. But, problem is like, if you're kind of relying on that, I mean, that's inflammation you can see and feel. What about the other inflammation that you're not able to feel in the arteries, in the brain, and other, why are you, allowing that to kind of resurface again? Why are you using that as a marker? So I think, I'd love to see people do trials where they cycle at different intervals, and we have better measurements on how to measure these things, whether it's inflammation markers, metabolic markers, mTOR, autophagy markers, and if somebody were to fund a trial, I think we can get those answers. But right now, nobody's done those trials. Right now, it's all just based on people's speculation, and people are free to speculate whatever, based on how they interpret the data or how they interpret, their understanding of the physiology. But it's too early to say.
Yeah, fantastic. Another thing that really came up quite a bit, we did see some early signs of significant improvements in women with these lower dosages, given the previous data. And we sort of touched upon this in mice, in non-human primates, suggesting different bioavailabilities in these organisms between genders. We in our bioavailability manuscript did not see such an effect. So a big point of, intrigue was, what could be influencing, and again, you might have to get your crystal ball out for this, what might be influencing the differences in effectiveness that we're seeing between men and women, and maybe does it give you any indications? Is there any pings on your radar about how this might influence, N of 1 dosing for individuals? Will there be a 6 milligram set it and forget it kind of dose? Or like you were mentioning before about tissue specific effects and things like that, do you see this moving in the direction of personalized prescription?
Yeah,
moving
it would
forward.
make my life a lot easier if there was a set it and forget it dose. If there, if 6 milligrams is a set it and forget it, that'd be great. Put everybody on 6 milligrams, and life would be rosy. And it's possible. I mean, it's we know from, or we seem to indicate from the Pearl trial that the 10 milligram unencapsulated or non-encapsulated, which is roughly about 3 milligrams encapsulated, seem to be too low for men, and seem to have some benefits for women. But the question is, would we see more benefits if we push the dose higher? I think we would. Again, to a certain point. At some point, it's going to flip. Just like a lot of things in life, there's like U-shaped curves or upside down U-shaped curves. So but do you have to have everybody kind of at the top of that curve, um, or is there enough leeway within the dosing? I would like to think or hope that there's enough leeway in the dosing so we can find kind of that universal dose for everybody. Um, you know, and like I said, my instincts or right now I'm thinking is between 4 and 8, and 6 kind of falls right in between that. So if I had to put money somewhere, I'd put money on 6 milligrams once a week for the encapsulated version, as kind of maybe the universal dose. Um, but I have seen clinically some people can't tolerate that high of a dose. They have some side effects. Too many recurrent canker sores. Uh, we've had a couple other weirder side effects. I can't remember off the top of my head, but they seem to resolve when we go down to the lower dose. And funny enough, they're all women. Um, I can't remember a single male patient that we've had to modify lower the dose on. Um, and that kind of again speaks to the data that we saw from Pearl. So right now I'm kind of sticking to 6 because that's where that survey, the 333 people survey, vast majority of people were on the 6 milligrams. Although most of those participants were also male, so we didn't get very much female representation there, unfortunately, just like Pearl. But like I said, I think I'm hoping that we'll see like a dose range, 4, 6, or 8. And then we'll have some better measuring tools on how to dial that in a little bit better for an individual patient.
Um, and I think another thing that comes up is that a lot of people use other interventions in conjunction. What are your thoughts about using some of those, especially glucoregulatory ones like acarbose or metformin at the same time?
Yeah, I mean, if you look at the mouse data on this, I think it looks like combinations work really well. I think they did a combination of metformin and rapamycin that seemed to improve response. They did combination of rapamycin and acarbose, I think. I don't know if they did the triple combination yet or not. But I know a lot of people are not optimistic about combinations. I'm actually quite optimistic about combinations. I think there's a higher potential for optimistic for combinations to help rather than not help. Some people say, oh, they might negate each other, but I've seen too many data sets that show that that you get a additive or maybe a synergistic, probably additive benefit with piling on multiple therapies. So that's why I'm very optimistic that it can only help with a much lower potential to harm when you're adding. I can't I think there's very, very few data sets that I've seen where adding is actually harmful. In fact, I can't even think of any off the top. I can think of at least a half a dozen data sets where adding therapies together is beneficial, but I can't off the top of my head, I can't think of a single data set that I've seen where it's the opposite. So I'm very optimistic about additive medications. What's the right combination for a particular person? That's nobody's even really looking at combinations, let alone looking to answer that. We have some hints from mice data, but it's I think it's too early to tell for males, for humans, especially human males and human females. I'd like to see rapamycin, LDN, and maybe an SGLT2 inhibitor like canagliflozin combination. I think that would be a very, very potent combination. But that's just speculation.
Well, that's a very intriguing realm of discovery right now. Um, but yeah, of course, first we have to continue gathering data that shows what the optimal, uh, you know, optimal use of rapamycin alone is, and move forward. But I think it's encouraging so far. Um, Another question that comes up, and this is a really hot topic in the geroscience or the longevity field now, is everyone's kind of coming up and iterating on a new biological age clock, right? And there's so many different types of omics measures, whether we're talking about epigenetics or glycans or whatever it may be, using those signatures to predict disease, to predict mortality, and these biological age clocks. I mean, so much so that in the commercial space, there's leaderboards of individuals, who has the youngest biological age, who's slowed down their biological age rate the most. So one of the things that came up is, how much emphasis would you put on biological age clocks as a marker of, how effective a gerotherapeutic intervention is, such as rapamycin? There has been this phenomena. People have reported anecdotally that their biological age is actually accelerated since taking rapamycin, while they haven't experienced any notable benefits or side effects either. And they're wondering at this crossroads, should I continue taking rapamycin based on these biological age results? And optimally, how do I interpret this and use it in a meaningful way moving forward? And what are your thoughts on that?
Yeah, I mean, I think we're still in the very early days of these bio age clocks of the various types. I mean, you mentioned several of them. Um, you know, um, uh, it's hard to say what exactly they're measuring. And some people have criticized some of the clocks out there that they're easily to manipulate. I think Peter Attia once, it was a while ago, but he was saying, if a clock is based on like your vitamin D level, all you have to take is, do vitamin D, go back, take a bunch of vitamin D, go back and get your clock measured again, and all of a sudden you're younger. Well, probably not. You've taken a bunch of vitamin D. Same with methylation clocks. If you look at like the TRIM trial, for example, the trial that looked at growth hormone, metformin, and DHEA, they reported this huge effect on the methylation clock. Um, you know, um, could there be something about one or more, one or a combination that kind of specifically targets the methylation process, and we're seeing a biased result? Um, I would say probably. I would say it's more likely that we're seeing an artifact of some sort there rather than, a true reversal or improvement in biological age. Um, on the other hand, multiple data sets have shown that rapamycin doesn't really seem to improve these methylation clocks. Um, but I think it's at this point it's kind of undeniable that rapamycin is doing something. With all the data sets that we have, human, dog, mouse, marmosets, it's I you know there definitely is something there. It's definitely doing something to the aging process. Uh, why isn't it showing up in the methylation data? Why didn't it show up in the glycan age, for example, that we measured? It's hard to say. It's hard to say. Um, you know, there might be more promise in things like metabolomics, or some kind of composite score looking at a bunch of these clocks together, because each one might be measuring a different aspect of aging, if it's measuring aging at all. It's really hard to say. I mean, I was very optimistic about the methylation clocks a couple of years ago. Then, like I said, very disappointing results with methylation clocks and rapamycin. Some people even reporting an acceleration of aging. I'm like, wait a minute, that doesn't match the clinical. When a test doesn't match the clinical picture, you either repeat the test or you throw it out. um, you know, um, so again, in this case, some of the methylation markers don't seem to match the clinical picture that we're seeing. Um, you know, if somebody has a better clock out there, where it does seem to match, um, you know, I'd be more than happy to kind of pursue that and see more data on it and see more studies done on it.
And I think, one of if the biological age clocks aren't it, what are your thoughts on which one, what biomarkers we should pay attention to?
Yeah.
If it's not clocks, what else, what is there that we should
Yeah,
monitor and focus
yeah.
on?
I mean, there's, if you take the X Prize approach, for example, the X Prize health span approach, I mean, they're looking at functional markers. I think that's a very reasonable approach. That's why we kind of did the SF-36 and the WOMAC for our Pearl participants, because I kind of suspected going into it that some of these, quote unquote fancier aging clocks would disappoint. And unfortunately I was right. I wish I wasn't right, but I was. So yeah, I think functional measurements is definitely a good good place. Um, I think there are plenty of unexplored areas as well. Inflammation, immune function, measurements of muscle function, measurements of cognitive function. Again, these are all function. I'm kind of harping a lot on function because I think that's something we can measure and tends to be important for people. Honestly, it doesn't really matter what your biological age clock says. It's how you're functioning. Are you able to do what you need to do or want to do? With that said, there's another question in here about, like, do we need to wait 50 years? Because it could take a long time to see some of these functional improvements, or to figure out whether, rapamycin is actually going to reduce the incidence of dementia or cancer, for example. It could take decades. So I think it is important that we continue. I don't want to necessarily throw out these biological age markers. I think they're important, but I think we still don't yet have a good grasp on what they're actually measuring, and how to use them to guide any kind of therapy right now. They're good as an educational tool, and if there's something really off, then it's probably an indication you're somebody's in major trouble. In terms of, like, really off in terms of like the delta between their chronological age and their biological age. If test comes back and it's off by a lot, then you really need to, like, sit and think about, like, what is what's going on. Do they have cancer? Do they have some kind of, like, autoimmune condition? And vice versa. If there's a big gap in the opposite direction, that and the clinical picture fits, somebody's doing everything they should be doing, good lifestyle, gerotherapeutics, etc. I think that's where it's useful. Where it's not so useful is, like, where you get small changes and you're not quite sure what to do with them. I think that's where it's less useful right now.
Fantastic. Thank you for that answer. And I think we're, sort of winding down uh,
Yeah.
in terms of questions and in terms of time. And you did mention X Prize, and I think that's an interesting, conversation you brought up in terms of just like the different endpoints they're using. They need to see improvements in immune health, muscle function, and cognitive health for an intervention to kind of stand a chance of winning this competition. And as a trial or a study that's assessing a gerotherapeutic, that's kind of the name of the game. You want to see it address multiple different organ functions, multi-morbidity. So you mentioned a few things. I know we're planning on following up in terms of some of the body composition changes we saw encouraged by the Pearl trial and some of the other data that we're seeing within our patients, dental records. Is there anything else? How would you weigh quality of life? Are there any other metrics that you're really interested in, like immune markers or
Yeah,
cognitive health that we
yeah.
think
I,
will
I
follow?
I talked about some of the ones I didn't talk about. There was, just this morning, somebody posted on one of the longevity doctor groups that I belong to that they're seeing plaque regression using carotid IMT. And that makes sense, because I think plaque buildup has to do with autophagy, or the lack thereof. So I think if you kind of help bring balance back to the autophagy mTOR system, then you could potentially see a regression of plaque. So that's something, I'd love to see a study on. I tried to propose it to a cardiologist I know, but he was too busy. But yeah, I mean, I think that's a very, potentially low-hanging and very impactful data set that we can unlock, if we had the right trial for it. What else? We talked about dental health, but also bone health. I mean, osteoporosis, osteopenia affects a lot of patients, especially female patients. I mean, it's less common in males, but very, very problematic in females. The drugs out there, sometimes you get very modest improvement, but at a cost, or you get no improvement at all, and you just kind of stabilize disease. I think rapamycin, again, with some combinations of our other drug therapeutics. So, we have a case study that we're writing up showing that we saw like a 5 to 15% increase in bone density. I think that's another area we could look at. Visceral fat, I haven't given up on that yet. Even though we didn't reach our primary endpoint for our trial, but I think I still suspect that we could see a difference if we look at higher doses, potentially longer periods of time. Maybe not longer periods of time, maybe just higher doses. So there's a lot. There's a lot. You talk about XPRIZE. I think they have like 20 tests or something that they want to run. I don't want to give anybody any competitors an idea, but I think rapamycin could potentially show improvement in pretty much every one of those functional measurements. So I think, and I wouldn't be surprised if there are people out there who are planning to incorporate rapamycin or a rapalog in their XPRIZE proposal.
This was, I think this was great. I don't know if there's any other, I think we answered a bunch of the different questions. There's always more coming in. So, I'd encourage anyone that's viewing this webinar to, engage with it, put your questions in here. You can email us for more questions. I know that there's a lot of interest in the topic. There's still a lot of ways to go. I know we're going to be, generating a lot more data moving forward surrounding rapamycin and its effectiveness. So, really exciting times. And if there's any other, final concluding words that we want before we close this out, we can go into it. Otherwise, I think this was really valuable.
Yeah. And I think, I'm very, very excited, very optimistic about the data set that we're currently crunching, based on our telemedicine platform patients. We've collected a lot of data there. It's not in the ideal format. So there is some wrestling we have to do with the data to get it to a place where we can analyze it properly. But I think once we start releasing that data, then we can actually come back and answer some more, potentially answer some of these questions a little bit more accurately. And also, I'm sure for every question that we'll be answering, it's probably going to be 3 more that pop up. So we're going to have to do this again. Probably stay tuned, maybe, I don't know, 6 months from now, plus or minus. So, send us your questions and we'll kind of put it on deck for the next time we do this.
Thanks so much, Dr. Z, Stefanie. I think this was really fruitful and looking forward to more exciting things to come moving forward. Thanks,
Okay,
everyone.
thank you very much.